Alright y'all, welcome to section 10, lectures 18 and 19. You guys just finished on exam 3, just want to say congrats, that was the hardest exam. Exam 4 is going to be much more chill. It's all about DNA replication, transcription, translation. It should be a lot more manageable from here on out. But yeah, just want to give you that assurance, and again, sorry for not posting. I can give a million excuses, but at this point, I just need to lock in. That's really it. So we're about to go straight into this. I'm just going to supplement basically the lectures with these. I know Professor Holmes is such a distinguished lecturer. He's very good at what he does. So honestly, like, you guys could solely just rely on his lectures and you're going to be just fine. But I'm just going to give you the extra supplement now, thinking about people who are studying for the MCAT. You know, for those individuals, there were a couple things that like Holmes doesn't necessarily cover in depth or that are briefly mentioned, but it pops up in the MCAT and they assume mastery. And it's just these extra supplements that I'm just going to kind of like toss in there, be like, hey, you need to know this and this and this. But yeah, let's get into it. Lectures 18 and 19. So lecture 18, we're going to talk about the very, very basics of DNA structure, topology, recognition, all that kind of stuff. And I'll hopefully be able to clear up some stuff. Hopefully slides are very comprehensive as well. So central dogma of molecular biology, as we all know, it's a little bit more complicated than this with all the advanced level classes, but we're going to keep a very brief overview of this. So first thing we have is DNA, then we have RNA, and then protein. So you know, DNA is obviously where we store all of our genetic code, and then a certain percentage of that's going to be expressed into RNA, and then RNA is going to act as this intermediate, like this adapter in a way, and be converted into protein eventually later on. So before we can move on to RNA, proteins, that's going to be much later down the line. We're going to talk about DNA specifically. So we know that DNA is an information molecule. That's where all our genetic code is stored. Some of it is expressed and some of it is not. And the ones that are expressed are called specifically genes. They could or couldn't be expressed, you know, it doesn't really matter too much. But basically, within genes, there are nucleotides that specify amino acid sequences of proteins. What this means is like you have an A, a T, G, combine three of them, you make a codon, and the codon corresponds to its amino acid. And then outside of these genes specifically that are not coded for amino acids, there are regulatory sequences for DNA replication and mRNA synthesis. All this means is that you may have a string of like ATTT or like the TATA box, for example, TATATA. Those are regulatory sequences that allow for DNA transcription factors to bind and then allow for DNA replication to take place. Those little regions of sequences may not code for amino acids, but they allow for DNA replication to happen at the replication fork. And then DNA is a polymer synthesized from DNTPs, so they're called nuclear triphosphates. So it could either be an A, a T, G. That's where we replace the letter N with the A or the T, the G, or the C, whichever one. And then for RNA, it would be the U. And then again, DNA is linked by phosphodiester bonds, and the basis available for hydrogen bonding and stacking. So this means that DNA, obviously we know that it's the double helix. They have hydrogen bonding and they have base stacking as well. That allows for that stability of DNA to occur, or for DNA to have a really high stability. And again, there's a 5-3 polarity. This is because the 5' end has the phosphate group, and that is negatively charged, whereas the 3' end only has the hydroxyl, which is not negatively charged at all. So it creates this polarity, this dipole moment within the DNA helix. And then here are the 4 bases for DNA. So we have deoxyadenosine, deoxyguanosine, deoxythymidine, and deoxycytidine. And what I want to emphasize here specifically is you should know specifically if it's a purine, and on the exam, you need to know how to recognize them, right? So you need to know, okay, if you were given this base right here, they'll say like, what is the nucleoside? Well, that's the trick question, because you wouldn't add the phosphate group to a nucleoside. Remember, the nucleoside is just the sugar and the base. Whereas a nucleotide, that's the phosphate, sugar, and base, so it's the 3. So you should know how to recognize which base is which, and then also if it's a nucleoside or nucleotide. I know in the exam we were kind of grading meticulously for that, so if like you wrote something very similar but it wasn't exactly the right answer, then we wouldn't have given you points. Same thing with this case. Like, you should know very well what the differences are. Okay, so moving on to DNA base pairing. So obviously you guys have heard of Chargaff's rule, which is just A binds onto T, C binds onto G. And I want you guys to know, this is very important for the MCAT, note how many hydrogen bonds are created between these different bases, right? So for the A and the AT has 2 hydrogen bonds, and then the CG has 3. Now, the more hydrogen bonds you have, the more stable that conformation or the bonding is, right? So the reason why I say this is because when we're talking about, for example, melting temperatures of DNA, where we can denature the DNA and split it and break those like hydrogen bonds and intermolecular interactions, right? Well, the more CGs you have, the more stable it is, the more it can resist changes in heat. So what I'm trying to say is the more CGs you have in the DNA, the harder it is to denature it. And this is a very important MCAT concept, because they may say like, oh, we created a new base that forms 4 hydrogen bonds. How will that affect the melting temperature? Then you have to say, well, more hydrogen bonds, more stable DNA, the TM is going to increase a lot. So I'm just bringing that up. This is just, again, supplementing. I'm kind of riffing off these slides. So sometimes DNA can be damaged, leading to like cytosine deamination, which just means that you get rid of the amine group from the purine ring, and that causes damage within the DNA because you can't form those hydrogen bonds like normal. So cytosine loses amine to form uracil, which is found, and we know uracil is only found in RNA, not in DNA at all. So we know specifically that this is bad in DNA replication sense. In mRNA, obviously a little bit different story, but we won't get into that. That's much more nuanced than this class needs. So DNA rich in uracil are subjected very easily to double-stranded breaks, and what this means is that it increases genomic instability and can be very harmful. So all you need to know is if cytosine gets deaminated into uracil, that is very bad. Okay, so the structure of DNA. So complementarity allows for easy replication of the DNA, and you end up forming a template and non-template strands. The DNA strands are anti-parallel. Again, what that means is we'll split the word up into two: anti and parallel. Anti just simply means, parallel just means that they're never going to intersect, but anti means that they're going in the opposite direction. And remember, DNA has that 5-to-3 polarity. So if it's, for example, we're talking about the bottom strand, the 5' is on the left and the 3' is on the right. Well, that's the direction. It's going from left to right. Whereas the other strand is going the opposite direction, right? The right is the 5 now, and this is the 3. So if we stack them together, they're going in opposite directions. That's what the anti-all means. The sugar phosphate backbones are on the outside, and the bases are stacked on the inside. This is important because obviously the bases would not be on the outside, or else how would they form those hydrogen bonds? It'd be very difficult and almost impossible for it to do so. And then it forms a double helix, as you guys know, and usually it's found in the right-handed helix state, if that makes sense. And again, it's stabilized by hydrogen bonds. The helix as well has a major groove, which is wide and shallow, indicated by this magenta box here, and then in the minor groove is just narrow and deep. All this means is it has to do with how exposed the nucleobases are to the outside extracellular matrix. Or the aqueous solution. So what I'm trying to say specifically is, as you can see here, we see a lot of the yellow, which this is the bases themselves. They're very exposed, right? And DNA polymerase can go in there and latch onto it, or transcription factors can latch onto this region right here on the top right. Whereas in the minor groove, as you can see, there's not that much yellow here. The bases aren't as exposed, so the DNA transcription factors that are going to bind onto there are the ones that are bound onto these phosphate groups. And an example of this, you don't need to know, this is like very much advanced level, are called polyamides or polyamines, one of the two, but specifically these will bind to the phosphate backbone specifically because they have a different conformation state. It's very advanced, you don't need to know that. I just was throwing that there to supplement it. But there are these two different regions in the helix itself. Okay, so now talking about the structural variations. So there are different forms of DNA. There are 3, A, B, and Z. Do I know why they picked Z? I had no clue. But Z is the most unstable. B is the one that is what we canonically think of DNA. And then A is the much more tight, but it is less stable, if that makes sense. So for the A form, we'll talk about specifically here is when we have DNA, RNA, or RNA, RNA helixes. And again, the one important thing that I will ask you guys to remember, if you can, is how many base pairs are per turn. So like A has 11, and this makes sense because it's tighter, right? It's much more compact, so it means you're able to do more turns in a single rotation. Or you're able to incorporate more base pairs within a single rotation, a single turn. That's the most important thing. And the B is the most stable, so it's a little bit wider. It's not as compact. And what this means is that we're going to have less base pairs per turn, and we're going to have 10.5. And then lastly is the Z, but this one specifically is left-handed. It is not right-handed. And with the Z, you don't really need to know the base pairs per turn, but I'm pretty sure it's like 12, if I'm not mistaken, or 10. I don't know. I don't remember. You see, you don't even need to know it. But all I want to say is that these are maybe in cells, but really they're mostly found in viral proteins that recognize the C form of DNA. And I kind of jumped the gun a little bit earlier on this in terms of DNA denaturation and ATCG bonds, but denaturation just simply means you're separating the DNA strands into two different sides, right? And this could be done by exposing DNA to higher temperatures or higher pH. There's a lot of ways you can manipulate it. This has to do with optimal stability, all that kind of stuff. But what I'm trying to say is if you take it out of these optimal conditions, the DNA is going to denature and split apart, and you will have two separate and not bound, like DNA strands that are not bound together. They're going to be separated. And so the TM simply just means the temperature at which 50% of the DNA is denatured, right? And the red curve simply means that the TM is much greater. Remember, in the blue curve, as you can see, the TM is much less. And what I mean by less and greater is the temperature at which DNA is 50% denatured, right? So the reason why I bring this up is because if it takes a higher temperature to break apart the DNA, that means we're going to have more GC-CG pairs, because again, it has 3 hydrogen bonds. Whereas if the temperature is less, right, the blue curve, as you can see, if it takes less heat to denature the DNA, then that means that there are less CG pairs that we have to break. There are like more abundance of like 2 hydrogen bonds in the AT pairing, if that makes sense. Hopefully that does. And again, this is because again, the red has a higher TM, which just means that it takes a higher temperature to denature 50% of the DNA. The only way this can happen is if we have more GC pairs, not more AT, because AT is the 2. And this is just a diagram showing what the process of denaturation looks like. And yeah, moving on to the next slide. So now we'll talk about RNA, specifically it's a single-stranded helix. It's not a double helix like we find in DNA. I will say that it is much less stable than DNA. Working with RNA in a wet lab setting, I've heard from friends who are in a RNA wet lab, they say that they have to be very meticulous about how they prepare it, how they handle their samples, because it often can degrade very quickly and promptly. And even in like the most mild like manipulations and conditions, it'll just degrade and you can't even use it. So something to keep in mind. RNA structures are more diverse though because of this, because it's single-stranded. It will often form these hydrogen bondings within itself, right? So I wanted to show you here is, for example, you can have a single strand where you have a bulge, for example, the A sticks out, and that's just because of stability issues. Then you have an internal loop. You can have a hairpin where, it forms those hydrogen bonds within itself in the RNA. So again, RNA have more diverse secondary structures, and they have uracil instead of the thymine, and can also have GU-based pairings, which are known as non-Watson-Crick-based pairings. You guys should know this exception in RNA is that the uracil can't bind to the G, and that has to do with just the instability of the RNA helix itself. RNase P simply just cleaves RNA into tRNA. So the precursor for tRNA, it cleaves that and it'll form tRNA. And then there are different types of RNA. There's mRNA, tRNA, rRNA, and we'll go specifically in the future lectures on what those specifically do. I just wanted to give a brief introduction to it in this slide. So now moving on to DNA binding proteins. So there are 2 types, right? There are sequence-specific proteins and there are non-sequence-specific. What I mean here is that the sequence-specific are going to bind onto the bases themselves, the A, the T, the G, the C, all that. Whereas the non-sequence-specific, those are going to bind onto the phosphate backbones. And the way it's able to, this is a much more advanced topic, but essentially the phosphate backbones are going to have a certain configuration, like spatial orientation, whatnot, like it has to do with spatial stuff, that allows for specific binding of these non-specific sequence proteins. And that's much more advanced, but that's usually how it'll work, is like the spatial of the phosphate groups. Yeah, and so some of the general functions of these DNA binding proteins are that they may activate or repress transcription, unzip DNA, etc., like all that kind of stuff. Moving on to DNA binding motifs. So there are a couple of DNA binding motifs. I'm just going to show you pictures of them in the next few slides, but for right now, I just want to talk about specifically just general overviews. You guys, I don't know if this will necessarily be emphasized in the exam, but I know for certain it will be on your MCAT. Like it was so annoying, and I'm very glad that like I went above and beyond when I took biochem and studied for it. And the MCAT, like I just pulled it out of my ass, and it was fine. It worked out. But I just want to kind of go over it because I don't want you guys to fall in the same situation like I did. So for the first one is the helix turn helix, and again, it's recognition of helix position in the major group. So it is entirely alpha helical. So the best example I can give you of this is the LAC repressor, where we have this DNA binding motif binding onto the LAC repressor. That allows us to control the LAC operon expression. Number 2 is zinc fingers. So zinc fingers are coordinated zinc ions, either for cysteine residues or two cysteine residues. It's either or. That's not really the specifics. You just have to know that there's a zinc in it. And the zinc ion has no function in recognition of DNA, but it is used in the thermodynamic stability of the protein fold. And that protein fold is the thing that's going to bind onto the DNA. And this is usually found in eukaryotic cells, eukaryotic transcription factors. The third one is the homeodomain, and it's widely conserved, and it has recognition helix inserted into the major groove of DNA. What that means is it'll bind into the major groove of the DNA, and it's often seen during development, where you're going to need it for a certain temporal aspect of it. That's why it's widely conserved, because development is ubiquitous across all species. We have to develop into the organism and fully mature. Number 4 is the leucine zipper. So we have the amphipathic helix, and the leucine residues stabilize dimerization. So leucine zippers, they're going to act like, you know how like if you have a jacket and you zip it up and like it's separated and then it comes together like that? It's exactly going to do that, but with DNA. And in the middle, it's going to be inserted, the DNA is going to be inserted in between the leucine zippers. And I'll show you images of it. Then the last one is the basic helix loop helix, and again, its basic residues bind to the DNA backbone, the phosphate backbone, because we know basic residues are positively charged, the phosphate backbone is negatively charged. That's something I want to point out for leucine zippers is that lysine and arginine residues bind in the major groove, and asparagine is crucial for recognition of the DNA. So I'll show you guys some photos of it. Now this is the helix turn helix again. So you see helix, then you see the turn, and then right here, sorry, and then you see another helix here again. And there is a DNA recognition helix that allows you to bind it to the major groove. Again, those bases are exposed, and that allows for the DNA-DNA interactions. Then the next one is the zinc fingers. So as you can see here, you have the zinc, and then basically it's not so much to say it's like a finger, it's just to say that the zinc allows for the stability of the protein to happen, and because those motifs, those zinc finger like functional domains, whatever you want to call them, those will bind onto the DNA itself. That's really the extent you needed to know. What I was asked on the MCAT, which of the DNA binding motifs are bound to DNA only? And it was zinc fingers was the answer out of the 4 option choices. Then next thing we have is the homeodomain. As I said, there's a recognition helix, there's a turn, and then a helix. This is just showing you pictures of it. Then the next thing we have is leucine zippers, which again, like I mentioned, it's like it's open and then it closes, and it's exactly where the DNA inserts itself into the middle, where it'll basically be cleaved. Essentially, is I think the function of it. Honestly, you don't even need to know that, but just showing you a photo of it. Then the next thing is the helix loop helix. So as you can see, we have a dimer forming helix, the loop, and then the recognition helix. And again, basic residues mediate the DNA binding. So here and here you would find basic residues on these proteins specifically. Okay, so now moving on to one of the more complex topics is supercoiling. So DNA is very long. We know that if it was like a single strand just fully elongated straight, like there's no way it could fit in a teeny tiny cell. So the way we do this is through supercoiling and then histones, but we'll talk about all that specifically. So supercoiling helps relieve strain, right? So naturally the DNA wants to be wound into a supercoil. So as you can see here, we have relax, which is 8 turns, strain to 7, and if we supercoil it, it allows for it to go back to the 8 turns, and it's back in the relaxed state. And again, this helps alleviate that strain, that torsional strain. Now, most DNA is underwound, right? It's strained. This means that there are less turns than the relaxed DNA would want. So supercoiling is going to stabilize this by forcing, again, forcing more turns to occur. And so underwinding facilitates strand separation is what I really wanted to emphasize. So whenever we have euchromatin, which is expressed parts of the DNA. Euchromatin is going to be underwound, which just means that the strands are going to be separated and replication can take place. But in cases where we have heterochromatin, parts of the DNA that are not expressed, those are going to be wound and they're going to be in the relaxed state, and so the strand separation is not going to occur. Okay, so now talking about topological linking number. So linking number, all it does is it quantifies supercoiling. So it specifies a number of helical turns in a closed circular DNA. It is undefined if the strands are not joined together. So for example, linking number here is 1, linking number here is 1. And here we have 6. We have 1. It's basically like the number of turns. So 1, 2, 3, 4, 5, and then 6. It's how it goes out and back in, if that makes sense. Hopefully that does. But so the way you calculate linking number, because they may give you a problem on the exam where they say, here are the number of base pairs. Now calculate what the linking number would be if there are 10 base pairs per turn, And so this can be used to define whether the DNA is underwound or if the DNA is wound, all that kind of stuff. So this is just the formula to calculate that. And then negative supercoils are left-handed, and underwound, which just means that it's a decrease in linking number. Remember, I want to emphasize this again. Underwound means that it is strained right here, and we have and the word underwound means that we have less turns, right? So here we have 7, whereas up here it is wounded up or winded up, whatever you want to call it, and it's 8 turns. So this one has more, but it is relaxed. We want it to have more, whereas this one has less and it's strained because of that. So I just want to emphasize that here. A negative supercoil means that's left-handed, and we're gonna have a decrease in linking number because it is underwound. We have less turns. Does that make sense? So then in terms of negative supercoils, you just have to memorize that negative is left-handed. And then for positive supercoils, and we know for the positive supercoils, when it is overwound, it is increasing the linking number. We're going from, the relaxed 8 turns to now like 9, 10. We're overwinding it, if that makes sense. And then positive supercoils, you have to memorize this, it's just that they are right-handed. That's all. In terms of memorization, just memorize negative left, positive right. That's all you really have to do. And then topoisomers are the enzymes that topoisomers are not enzymes. That's topoisomerase. Topoisomers are 2 forms of circular DNA that differ in a topological property. Example as linking number. So if you have 2 DNA strands, one is overwound, one is underwound, then those are topoisomers. And topoisomerases change the linking number of the DNA to be wound, overwound, underwound, and whatnot. So there are 2 types. Specifically type 1, there is type 1 topoisomerases, which change LK in increments of 1, and it cleaves 1 strand of the duplex DNA, and it can relax both positive and negative supercoils. And it does not utilize ATP hydrolysis to achieve this. So for example, here's just a diagram demonstrating this. As you can see here, we have topoisomerase 1 binding to the DNA. It cleaves the 1 strand. It doesn't really matter specifically which one. You don't need to know that. Just know that it cleaves 1. And what it'll do is it'll basically pass the other strand that was left alone through and then reconfigure or reconnect or religate, I think that's the better word, the strand that was cleaved, and now you have basically increased or decreased the linking number depending on which one you had in the first place, if you had a positive supercoil or negative. Now let me ask you guys this. If you have a negative supercoil, we know that it is underwound, right? Meaning that we want to wind it more. So if we were talking about topoisomerase 1, it would increase the LK number. It would wind the DNA more. Whereas if we had a positive supercoil, we know that it is an increased LK. It means it's overwound. We need to unwind it. So what we're going to do is we're going to basically unwind it by 1, if that makes sense. Hopefully that second part does, but essentially we're trying to take it from 9 turns to 8, so now it's relaxed again. We're taking it from 9 back to 8 here, and 8 is the relaxed state. I'm just using that as an example. Okay, so now talking about type 2 topoisomerases. So type 2, it's insinuated in the type name, it changes alkane increments of 2, and basically it cleaves both strands of the duplex DNA, and in order to do so, it needs to use ATP. So that's the one thing you have to know, is that type 2 uses ATP, type 1 does not. So in this instance, right, it can relax both positive and negative supercoils, and it can introduce negative supercoils in prokaryotes only. And some examples are topoisomerase 2 and then DNA gyrase. And so I'll walk you guys through this mechanism. You don't really need to know it in depth other than just for your own information's sake. So as you can see, DNA is going to bind into the end gate, and what the end gate is going to do is basically close it and then keep the DNA in there. As now, when the ATP is introduced, it will basically cleave apart the 2 strands and force the unwinding. In this case, I think is what it's showing. Second link, yeah, it's passed through the broken DNA. And then again, the broken DNA is religated within the C gate in the bottom one, this blue portion of the topoisomerase 2. And what ends up happening here is now the DNA is religated, and now we have our DNA either unwound or wound much more, depending on if you have a positive or negative supercoil. Now, that was supercoiling. Now let's talk about nucleosomes, right? So nucleosomes are DNA being packaged around these proteins that are circular, and the proteins, the histones themselves, are often positively charged. Why is that? Because they interact with the phosphate backbone. It would make sense for the histones to interact with the inside of the DNA with those bases and disrupt the hydrogen bonding. We wouldn't want that. So it's going to interact with the phosphate backbone to maintain the base pairing. And so it's very easy to think that nucleosomes are going to have a high % of lysine and arginine residues, right? And it makes sense because the DNA backbone is negatively charged. Something you need to know is that the nucleosome particle, the core particle, is composed of 2 isomers of the histone subunits, if that makes sense. So there's 2 H2As, there's 2 H2Bs, there's 2 H4s, 2 H3s, to make a total of 8 subunits total. And again, it's found in chromatin of all eukaryotic cells, and the packaging of DNA in nucleosomes facilitates the supercoiling and allows for compact DNA and euchromatin or heterochromatin to form. So that's lecture 18. Now moving on to lecture 19, DNA replication and repair. So there are different possible models for replication, right? So there's semi-conservative, conservative, dispersive. As we know, the correct model is the semi-conservative, where you take your two original parent strands, you split them apart, and from there what you're going to do is you will have your two separate strands accordingly. And whereas the other ones, they're not really, we know that they don't work, and that's because of the Neselson experiments, or the Meselson-Stahl experiments, which I'll explain in a little bit. This is just for your own information, and honestly, it's going to be tested on your MCAT, so I thought I should go into this. But conservative just means that, like, out of your first replication, one is the original parent strand, and the other one is the new. And then semi-conservative is like a 50-50 split of the new, and of the two new strands, so you're going to have 50% parent and 50% new daughter strands in both of the DNAs, whereas dispersive is just like a random quantity. Different parts of the DNA are dispersed within the first replication DNA fragments, or DNA helices. So we'll go into them specifically. So the Meselson-Stahl experiment, what this did is we have one DNA that, or grow DNA in heavy nitrogen. So this isotope of nitrogen is going to be much heavier, right? And so we have a band shown here, and this is our original parent molecules of DNA. They're going to be much heavier just naturally. Now what we're going to do is we're going to transfer this DNA band, and we're going to put it in a new medium that is going to have N14 now, the regular nitrogen, not the heavy isotope. So we're putting the heavy DNA into just regular conditions, right? And what we're going to do is we're going to allow for one round of replication, right? And what we're going to do is we're going to see what ends up happening to the bands, right? If this was a conservative model, right? If we were to go back, if we were to form the original DNA parent strand that was untouched, and then the newly replicated daughter strand, what would end up happening is, if that was the case, ignore the band right here, but if the conservative model was correct, we would have two bands after the first replication. That's because we would have the parent strand that was untouched. It's going to be the 15N nitrogen, if that makes sense, the heavier one. It's going to be on the bottom after we centrifuge, and the 14 nitrogen of the daughter DNA strand. Again, this is the 15, this is the 14. We would have two separate bands after the first replication. That's if that model was correct. But what this experiment found was that there was a hybrid of it, right? Instead of forming two bands, we formed a single one in the middle of it. And so as you can see, we have 15N and 14N nitrogen being incorporated into the DNA daughter molecules, or daughter helices, as someone would like to say. So we know that it is the conservative because, again, we form a 50-50 split of our new daughter molecules. 50% is the parent, the blue, and 50% is the newly replicated, the daughter. And remember, the daughter DNA strands are gonna have the 14N, and the original parent are gonna have the 15N, because the parent strands aren't changed at all. And then the reason why dispersive is not true is because with dispersive, we would expect different bands to occur because we don't know which ones are being traded or which ones are not. We don't know which strands are going to be replicated, which one is the old one per se. So it's very confusing. And with dispersive, we would just expect weirder results. So the only plausible ones were conservative and semi-conservative. Okay, so moving on to DNA synthesis. We know DNA polymerase, which is the enzyme that catalyzes DNA synthesis, requires three different things. The template strand, makes sense. I mean, you just can't replicate it out of the blue. RNA primer with 3' OH, and DNTP substrates. So DNA polymerase, again, catalyzes the nuclear attack by 3'. Okay. So the reason why DNA polymerase requires the RNA primer is because it has to do with the first step of the reaction, the building blocks. Remember, we use RNA primase in order to start DNA replication. So something to note. In terms of this, so DNA polymerase catalyzes this reaction where we have a nucleophilic attack by the 3' OH. Remember, 3' OH can be deprotonated, and it'll essentially attack the 5' end of the newly incoming DNTP. And the DNTP has the 3 triphosphates. Again, it has that electrostatic repulsion. It has that energy to power this reaction through. So in a way, you're alleviating the strain and the electron repulsion from those 3 phosphates here. As you can see on this diagram, this 3' OH, or now it's deprotonated into an O-, this carboxy anion, or this oxy anion, apologies, it's going to attack the alpha phosphate group, and then from there it's going to form a phosphodiester bond and release this pyrophosphate. Remember, we've talked about pyrophosphate all this time. It's going to immediately split into two separate inorganic phosphates, and that's what drives the reaction forward and powers it too. And the snug active site, so this is what talking about the active site of DNA polymerase. It can only accommodate correct base pairs, Watson-Crick base pairing, and the only exception is the Y family polymerases, which are much more prone to error, and we know it's been studied in the literature. And so this is just something you should know, is that the active site is it's very intentional in terms of what can fit in and what cannot. What can fit in are the correct base pairs, what cannot are the non, and that's where we have DNA repair mechanisms come in. And the key active site feature is that magnesium is a necessary cofactor coordinated with a highly conserved asparagine residue, which forms this, and I use this vocab term, bidentate interaction with the asparagine spartate residues, with the carboxylate anion here. Okay, so now talking about DNA replication, there are three major steps. There's initiation, there's elongation, and termination. And just a little note, transcription and translation also have those three initial steps as well. So just don't get the mechanisms confused, keep them organized. DNA replication, just read the name. DNA replication is going to replicate DNA. Transcription, we're going to take DNA, convert it to RNA. It's like transcribing something, right? You listen to music and then you write music notes on it. You're transcribing it from one form to another. And then translation, that one just means that we're taking our music that we made and now we're translating onto the music that we're playing onto the instrument and whatnot. Okay. So just want to give you some background information about the DNA polymerases. So as you can see here, DNA polymerase 3 replicates the genome. It has the highest processivity, and processivity just means how long enzyme can hold onto DNA without falling off. That's all it means. So like once DNA polymerase 3 is bound to the DNA, it's not going to come off until all the gene is completely replicated. Does that make sense? Or until the DNA is replicated. And this makes sense. Like we wouldn't want to just replicate half of our DNA and then the other half is not replicated. That'd be very bad, and I'm pretty sure you can see why. So all of them have 3 to 5' exonuclease activity for proofreading. And all this means is it allows for removal of one base at a time. Like I said, the active site is very snug, and if it doesn't fit, it's going to immediately have this exonuclease activity, aka it's going to be able to repair the bad base pairing, the incorrect base pairing. And so incorrect nucleotide insert approximately every 10 to the 4th, 10 to the 5th base pairs. And that's just because DNA polymerase doesn't have 100% efficiency, right? It's not going to be perfect. And so with this exonuclease activity, it's an extra thing added to DNA polymerase. I'll show you the whole structure in a little bit. But with this extra structure, it's going to improve the accuracy by 10 to 100-fold. And all this means is that the exonuclease activity will rip that base pair apart, it'll correct it, and then reinsert itself back later, and with the corrected DNA helice, DNA base pairs. And then DNA polymerase 1 has 5 to 3' exonuclease activity, and it removes the primers and allows for removal of many bases at a time. And so these are just the two that you really have to know, DNA polymerase 3 and DNA polymerase 1. And then also I want you to note on this table is that the processivity, right? DNA polymerase 1 goes for about 3 to 200 nucleotides. DNA polymerase 3 goes over 500,000 nucleotides, so something to note as well. So now talking about DNA replication initiation. So in terms of initiating DNA, it involves specific sequences and proteins. There is the DUE, the DNA unwinding elements, and so again, which type of base pairs are going to facilitate this? It's the AT, because again, they have 2 hydrogen base pairs and they don't have 3 like GC. GC would be very hard to break apart. To remember, I kind of gave you guys a little hint on this slide right here, but strand separation involves like DNA denaturation right here, right? We basically but we disrupt the hydrogen bonds between the base pairs and allow for the strands to separate and open up more, so DNA polymerase, that initiation stage, can come in and start this replication process. So that's the first thing. And DNA A recognizes the ORIC sequence where the duplex split. Again, it's where the strands separate, and that's the origin fork or origin of replication, sorry. And then DNA B is helicase, and it unwinds the DNA even more. It's kind of helping break apart all the DNA strands from the start. And then DNA gyrase relieves the strain generated by unwinding. So remember, when the DNA unwinds, it's going to become underwound, and that means that there's less base pairs per turn. There's going to be from 8 to 7, right? These are just made up numbers, but it's going to be underwound, and that creates torsional strain. The DNA gyrase is a type 2 topoisomerase, it's going to unwind the DNA and alleviate, or sorry, it's going to start winding the DNA and alleviating that strain. And then lastly is the SSB, which is just, man, what is it called? It is the single-stranded DNA binding proteins. And what this is, all it does is it's going to bind to the single-stranded DNA and stabilize it. The DNA wants to come back together. It does. But the SSBs are going to bind onto the single-stranded DNAs and keep them separated. That is their function. And then this diagram right here is just showing you kind of what I was talking about in the previous steps. Realistically, you really need to know these specific proteins that are binding to the sequence, the DNA unwinding elements, which is AT-rich. That's really all you have to know. This is just showing you a picture of it. If this is complex or too much, I wouldn't stress about it. So moving on to elongation, right, the second part of DNA replication. So it starts at the origin of replication, if we're talking about circular DNA, and the primase is going to lay down these RNA primers. Again, it's not DNA primase, whatnot. It's not DNA primers, it's RNA. And at the origin for the leading strand, and there are two areas, because obviously one strand is the leading strand, that one just replicates continuously, and then we have the lagging strand, which it's almost like sequential, right? These are called Okazaki fragments that we make, and there's like a little special step, and we'll talk about it. DNA B helicase unwinds the DNA. It, again, proceeds, it goes ahead of it, all the entire structures and unwinds it. And then we have DNA polymerase 3 adds nucleotides. SSB stabilizes the strands that are basically unwound and separated. And then DNA gyrus relieves the strain by unwinding the rest of the DNA, supercoiling. Okay, so now talking about the lagging versus leading strands. So one thing I want to emphasize is this bottom right diagram right here is the direction in which the replication fork is moving. And it's designated by these big arrows, right? So I wanted to emphasize this because we know DNA is replicated in the 5' to 3' direction, always. Always. So I really want to emphasize this. The DNA, the newly synthesized strand, the new strand is going to be synthesized always in the 5' to 3' direction. So what does that mean about the template strand? In which direction does DNA polymerase read, not replicate, read the template strand? From the 3' to 5', because that part of the DNA helix, that antipart, it's going to retain that. So in this case right here, right, the replication fork is moving to the left. And we're talking about this top left, like, quadrant, right, per se. This top left one, we see that it is the leading strand, and that is because the 5' to 3' direction matches with the direction of the replication fork. The easiest way, if on an exam they tell you to write one out, like entirely, like this replication fork, directions of the strands and everything, just first write the parent strands. The 5' to 3', they're going to be opposites, right? Then from there, whichever is your leading strand, that's going to follow the direction of the replication fork. So your 5' to 3' is going to be in the same direction as the direction of the replication fork. And then you have the lagging strand. And the reason why I want to say this is because the lagging strand, it's being synthesized in the opposite direction of the replication fork opening. So in this case right here on the bottom left diagram, acquainting you, we know the replication fork is opening to the left. But at a certain point, it opens up enough, and we put in our RNA primer, but it's going back in the opposite direction, right? It's not following the replication fork opening. So now as this continues to open, you're going to have these open spots right here that are not going to have an RNA primer, and that's where the primase again has to come in and lay down the new RNA primer. So the lagging strands are always trying to just keep up with replicating because it's going in the opposite direction of the replication fork, and it creates these fragments known as Okazaki fragments. And so that's something you have to keep in mind, is like you can think of this as like, I like to think of it as 4 quadrants. I don't know if you're seeing the same thing. If not, just ignore what I'm about to say. But I divide this into 4 quadrants. One of them, basically 2 of the quadrants are going to be the leading strands, and then 2 of them are going to be the lagging. And that's just because the direction of the replication fork is going to open in the opposite direction, and whichever strands are following the direction of the replication fork are going to be the leading strands. And there's 2 of them, so there has to be 2 of the strands that are leading. And then in consequence, there's going to be 2 strands that are lagging. And hopefully you guys saw that. If not, just ignore what I just said. That's just the way I think about it in my brain. But yeah, so leading strand is continuous, lagging strand is discontinuous. Yeah, and that's pretty much all I want to say, is that it's bidirectional, meaning that you're going to have 2 of these in each direction, so it makes technically 4 different processes. I don't know. I just think of it as quadrants. If that sticks, it does. If it doesn't, just ignore it. Okay, so moving on to the next thing. So continuing in the terms of this, in terms of DNA elongation, so DNA polymerase 1 removes these RNA primers, right? Remember, the processivity is about 20 to 100 nucleotides, and these nucleotides could be RNA primers themselves. So it has the 5' to 3' exonuclease activity, which removes these primers. And also, it actively fills in these gaps with proper dNTPs, changing it from RNA to DNA. And then DNA ligase is going to connect the separated strands, right? So as you can see here, it seals the backbone, and it forms these phosphodiester bonds. So you have the adenylation of the enzyme right here, as you can see. So this is the enzyme, this is DNA ligase, and then it gets adenylated with adenine ribose and all that stuff. And then from there, it's going to activate the 5' phosphate. And you don't really need to know the mechanism per se, the arrow pushing. I'm just showing you in this diagram. But essentially, the enzyme is going to allow for the phosphodiesterase bond to occur and to form. And again, you need to have this adenylation in order for this reaction to occur right here, this step number 2. As you can see, we have enzyme AMP. We no longer have enzyme AMP anymore after the reaction is done. So you're passing on this adenylation group onto the DNA replication, onto the newly replicated DNA strand itself. And then from there, DNA ligase will basically seal it all up. And again, the enzyme will alleviate the AMP from the newly replicated DNA strand, and you end up forming that phosphodiester bond and that sealed DNA. So you don't really need to know this mechanism. You just need to know that the enzyme can be adenylated, and then it allows for the phosphodiester bond to form, and then now you have a continuous piece of newly replicated DNA. Okay, so DNA replication, and this is one of the slides I wanted to mention about the structure of DNA polymerase 3. So DNA polymerase 3 has these key features. It has a beta clamp, which is intended to ensure the DNA polymerase does not fall off the DNA strands. This is why it has high processivity. It is not by coincidence. It is because it is purposefully made to have high processivity. So the leading and lagging strand synthesis is occurring at the same time. They're not occurring sequentially or one after another. It's at the same time and simultaneously. And the lagging strand synthesis is discontinuous, which is why it requires steps B and D and constant loading and unloading of the beta clamp, which requires ATP hydrolysis. So I would take the time, go through this very slowly on your own time. I'm not going to go into it because you don't really need to know like this whole step. I'm just telling you why DNA polymerase has that high processivity, why it can stay bound to DNA for so long. And it's because of that beta clamp. I would really walk through this very slowly in terms of this bottom part right here, as you can see, and watch how the beta clamp will shift and then cause DNA polymerase to synthesize the rest of it. I think that's very crucial if you want to get a better understanding. But I won't go into it in this video. So now moving on to the last step, DNA replication, the termination. The replication fork stops in the terminus region into these termini, like terminus sequences, stop sequences, whatever you want to call them, TER. And when it stops here, you're going to form 2 duplicated chromosomes that are catenated, which just means that they are entangled. It means that they're not going to be already like separated. They're going to be entangled, and you need an enzyme to untangle them, to separate them fully. And that's going to be DNA topoisomerase 4. DNA topoisomerase 4 is going to take the entangled chromosomes right here on this right diagram, and it's going to separate them into 2 separate strands. And it's exactly that separation that will cause now these chromosomes to be fully apart. And so replication in eukaryotes, it's a lot slower and more complex. There are 5 nucleotides per second in a eukaryote, 50 nucleotides per second in E. coli. As you can see, it's a lot slower, it's more complex, there are more things playing into it, but just want to say that eukaryotes are more complex than prokaryotes. That's really it. Chromosomes in eukaryotes are long and linear. I want to really emphasize the linear rather than circular. And they require multiple origins of replication to replicate efficiently. So with like a plasmid, for example, circular DNA, you probably only have 1 replication fork and it makes a nice little loop. And that's because it happens more quickly, it's not as complex. Whereas in eukaryotic DNA, we have multiple replication forks and they all split in their own directions, and you are able to replicate DNA much more efficiently because you have multiple areas being replicated at the same exact time. So again, for replication eukaryotes, initiation requires 2 steps. Formation of the pre-RC replication complex. CDK enzymes are usually off during this time. And then the second step is coordination activation, where the CDK enzymes are turned on, and it involves ATP hydrolysis. Remember, the beta clamp needs ATP hydrolysis in order to attach itself onto the, onto the DNA. Like, that's just an example. And then note, cyclin and CDKs are important cell cycle regulators. It would make sense that CDKs are important for DNA replication, again, which occurs before mitosis in the S phase. So in terms of eukaryotes, we remember how I said they were linear. Well, at the very ends of these DNA, not circular DNA, so these long linear chromosomes, at the very ends we have these things called telomeres. And telomeres, they don't loop around. They're just linear and they end off as it is. And so we need a special kind of enzyme to replicate it, right? And so telomerase fixes this problem, right? One thing I want to emphasize is that, or one thing I want to clarify is that telomeres fix this problem with the linearness and the fact that it doesn't loop. At the very ends, we have repetitive nucleotide sequences which shorten after each replication cycle. So that's why we talk about telomeres always shortening as life progresses. And we don't really care if we lose these nucleotides because they don't actually code for proteins, so they're not really useful. They're just like caps for the ends of DNAs. And it's once we lose those caps and we start going into the actual DNA sequence where we start to see like detrimental effects, aging, all this negative stuff. All these cellular processes start to have maladaptations to it. So telomerase is the enzyme that keeps the telomere caps as long as possible. So telomerase synthesizes DNA from an RNA template, and it's called a reverse transcriptase, and the template is part of the enzyme. So as you can see here, we have telomerase, we have a template that's RNA, and it's going to essentially act as a reverse transcriptase, taking this and essentially elongating the DNA sequence, as you can see, and then it inserts an RNA primer. DNA polymerase fills in the intervening gap, and DNA ligase seals the final nick, which, as you can see here, we have the RNA primer, DNA polymerase makes the strand elongate, and then from there we have DNA ligase forming. So I want to emphasize really quick the reverse transcriptases. All they do is just take RNA and convert it from RNA into DNA. So it's going in the reverse direction from DNA to RNA. So hence reverse transcriptase. And so it's RNA-dependent DNA synthesis. There are two examples. HIV, it's a retrovirus since it uses a reverse transcriptase and injects into the cells. And then line 1, which is a retrotransposon, which we won't go into that in depth, but these are just two examples. So now moving on to mutations, right? Mutations can be silent, deleterious, advantageous. They can do a lot of things. We don't really know. That's why they're called mutations. So silent just means that there's no effect on gene function, no change in amino acid sequence, despite the nucleotide changing itself, right? For example, we can change it from a T to a C, but it doesn't change the actual amino acid sequence. And that's because amino acids, the translation part is degenerate. It's a degenerative code. And we'll talk about that in the next few lectures in a few. We'll talk about that in the next few lectures. So then there are base substitutions. There are which are basically called transitions, transversions, base insertions, base deletions. So we'll talk about each one. Transitions just means that we're changing the base from a purine to purine, or pyrimidine to pyrimidine, and they very frequently lead to synonymous changes in the code, meaning that they do not change amino acid sequence in the protein. They're basically silent at this point. So as you can see here are some transitions right here where we convert AT to GC, or TA to CG. So essentially like it's silent mutations. And then transversions are conversions of purines to pyrimidines. So you're changing the ring size from a 2-ring to a single ring, right? And so it can significantly change protein sequence, and this is less common. This is where, if we have a transversion right here, it could lead to very deleterious like adaptations, mutations. And then we have base insertions, which a new base just inserts, and then base deletion, we just take out a base completely and shift the reading frame. These are more common in terms of which ones cause these deleterious mutations, but for the most part, these are the two main that you have to know, and then these two are kind of some newer ones. But this diagram should show that much more in depth. Okay, on to the next thing. So deamination. So deamination just means that we're removing the NH2 group on the carbonyl, we're removing the NH2 and forming a carbonyl in its place. So as you can see here, we have cytosine, we deaminate the top, and we form this carbonyl, which means it's uracil, right? So this is how you can get uracil in a DNA molecule, right? And deaminating agents can induce conversions at high levels, meaning that like nitrosamine, sodium nitrate, sodium nitrite, nitrous acid, like precursors, can cause this deamination to occur in the first place. So here's another form of mutations, depurination, alkylation. So it can happen spontaneously, and it's due to alkylating agents. So depurination, as you can see here, involves this. Depurination means that we're basically getting rid of the purine group from the sugar and the sugar and the phosphate group. So as you can see here, we have this connection, this base sugar bond right here, and with the addition of water or alkylating agents, we can break that bond and basically create a apurinic residue, or called abasic in other words, and our free base. And so alkylation can change base pairing properties. So for example, the O6-methylguanine will base pair with thymine instead of cytosine, and this will cause mutations in subsequent replications, and it'll change to AT bases essentially. So this is another, you don't really need to know the specifics of this. You just need to know that depurination just means you lose the base of it. And then alkylation just means we're adding a methyl group to one of these hydrogen bonding residues, and that changes also the base pairing as well. So all this is just saying is that base pairing can happen incorrectly, and it's because of these two mutations. So UV radiation, this is a special one where we add UV energy or UV light, and UV is much more, it has a higher frequency, it has higher energy, and this can cause thymine dimers to appear, to form. And so as you can see, the kink, when the thymine dimers form, they're gonna basically form from up and down base pairs, and it creates this kink in the DNA. And the kink can lead to blocks in replication and transcription because it'll literally force off the DNA polymerase II. And so again, this is caused by the formation of a covalent bond between two adjacent thymidine residues. So as you can see here, the formation of these two, and it creates the kink. DNA polymerase can be pushed off, essentially forced off of the DNA replication. So let's say we have one of those issues. How do we repair it? So there are 4 types of mechanisms. There's mismatch, base excision, nucleotide excision, direct repair. We'll talk about each of them. And I would just be familiar with which repair mechanism is usually used for certain types of damage, which we will clarify with this. So in terms of mismatch repair, right, it corrects replication errors. So like, for example, methylation is a form of epigenetic regulation, differentiates the old strand, the methylated ones, to new strands, which don't have the methylation in the first place. And there's a small window of opportunity before new strand becomes methylated so that we can repair it, right? So the process is, where it's a very long complex. It's basically this picture, but we'll break it down into simple terms. So the mut-L, mut-S binds to the mismatch, right? These proteins. So as you can see here, it's designated by these little like triangles. So we have the mismatch base pairs. The mut-S, mut-L are going to bind onto those, and it's going to form this complex. It requires ATP hydrolysis to bind onto it, and then once this complex forms, the mut-H is going to find the methylated site, right? So we have these two form, and the mut-H comes in. It's going to form this like loop essentially. So we have the mismatch base pair on the top now, and we're going to form, the mut-H is going to recognize the methylated components of the DNA strand, and what it'll do, it'll cleave the unmodified strand where there's no methylation marks, and it'll basically cleave the strand at where the methylated is. And it's not going to cleave the parent strand, because the parent strand is the one that's methylated. The daughter strand is going to be cleaved. So as you can see on this arrow right here, you see this little portion right here where it's cleaved now. And so what ends up happening here is that we have this exonuclease activity where we degrade the mismatched base pair, right? The parent strand is fine. It's just the daughter strand that forms that mismatched base pair in the first place. So it's going to cleave it, and then from there we have DNA polymerase III do the replication again and fix it. And so that's basically what this is saying, but in words. So again, it uses this process, mismatch repair, uses ATP. It uses a very like complex like enzyme formation complex, whatever it's called, the mut-L, mut-S complex. And you just need to know that it basically cleaves the DNA. And once it cleaves it, it has exonuclease activity. And once it does that, the DNA polymerase can replicate the rest of it. But this requires a lot of ATP in the first place to do this process. So now talking about base excision repair. So this repairs damaged bases like uracil and DNA due to cytosine deamination. And so what ends up happening is the process is that DNA glycosylase cleaves the N-glycosyl bond, meaning that the bond that combines the base with the sugar is going to be cleaved, right? And then AP endonuclease removes the sugar from there. And so as you can see here, from this to this, it's going to cleave that phosphodiester bond between the sugar that's abasic, meaning it doesn't have a base, abasic. So you can see here, DNA polymerase I is going to go from this end of right here, the 3' end that was just cut by the AP endonuclease, and just synthesize a new strand of DNA, new DNA. As you can see here, the DNA polymerase I, new DNA is designated by the red, and then from there, there's going to be a nick that's still created, and ligase is just going to seal it right there, and that's how you have base excision repair. So we just remove the base, then we remove the sugar, then we allow DNA polymerase to replicate the DNA, and then from there, we seal it with ligase. That's this one specifically. And then nucleotide excision repair, this is the next one. So this removes bulky lesions. So like, for example, pyrimidine dimers from UV irradiation, it forms a DNA lesion. And so from here, what we're going to do is we're going to use an XC nuclease, which is just an excision endonuclease, and it makes 2 cuts around the affected area to remove it. So as you can see here, we're looking on the left side right here in E. coli. As you can see, we have a DNA lesion. We're going to go upstream and downstream of that and basically cut it off. Like, we're going to do a very like non-precise cut, if that makes sense. And when we cut this, so once we make these cuts, right, it's still bound to the DNA because of the DNA base pairing, right? well, the helicase is going to basically disrupt the base pairing, and that's going to get rid of this damaged DNA. And then from there, DNA polymerase 1 comes in and basically replicates up until we get to the neck where it's not going to fully finish the replication. DNA ligase comes in and finishes it off. So the difference between the base excision repair is that we're literally removing the base and then the sugar, and then this one we're just like, we're not going to focus on the one base, we're just going to take off the whole DNA itself. So that's the difference between these two. And then in terms of direct repair, this is going to repair that base like directly, and so we're not removing any bases or nucleotides, we're literally repairing it. And so this is metabolically expensive to do it, to keep everything intact while also repairing it at the same time. So it's going to use a suicide enzyme, meaning that this enzyme is never going to be able to be used again. It's going to become inactivated. And so we use up one protein per repair. So protein cannot be used again. Again, that's what suicide enzyme means. And so in this example, we have a methyltransferase basically uptaking the methylated guanine residue, and this enzyme will now become inactive and never be able to be used again, but it'll repair the guanine base, and there we go, we just have direct repair from that. So again, if we have the guanine alkylation, this is where this direct repair could come into play. And so, sorry for like talking really fast, going through it, but I know Holmes does a much better job in terms of explaining all this, so this is just like a supplement in terms of what you need to know for the MCAT. I will get started on the next two videos as well and post those very shortly.